All right, so it is my great pleasure to introduce a brilliant scientist and mentor of mine, Jeff Kelly, for his seminar today. I had the privilege to work with Jeff from 2009 to 2012 as a postdoc in his lab at Scripps before I came to MIT. Jeff got his start doing a bachelor's in chemistry at SUNY Fredonia, and then a PhD in organic chemistry with Slayton Evans at UNC Chapel Hill. And then after that, he was with Emil Thomas Kaiser at Rockefeller. Started his career at Texas A&M before moving to Scripps Research. What year was that? 99, right around 2000. And has been at Scripps Research Institute ever since. He's the Lita Annenberg Hazen Professor of Chemistry and Chemical Biology. Jeff has, I think, a really amazing track record across the board. He's mentored and trained hundreds of postdocs and graduate students, and you can find them all over research universities and pharma and biotech and government. Lots and lots of science by the folks that he's trained, in addition to all the amazing science from his lab itself. He's been dedicated to service, both at Scripps as chair of chemistry, chair of molecular and experimental medicine for a decade, leadership roles at Scripps, dean for the graduate program, and also to the broader scientific community, including serving as the president of the Protein Society. So there's a super long list of papers and very long list of awards that go with Jeff's CV. So I'll just mention one of the most recent, which was the $3 million Breakthrough Prize in Life Sciences in 2022 for Jeff's work on the Transthyretin amyloidoses. And I think you'll hear at least a little bit about some of that today and more as well. But I can tell you the Transthyretin story is one of the truly beautiful stories in the literature, going from very fundamental biochemistry to a deep understanding of the genetics and the biology, all the way to design of small molecule drugs. He's one of relatively few academic PIs that actually invented and created the drug that is now in humans in his lab. And has started various companies. That was at FoldRx, is where that drug went. It was acquired by Pfizer, and I think I heard that it was $700 million in revenue last quarter. So it's looking like a multi-billion-dollar a year drug for these amyloidoses, but far more important, it's really having a tremendous impact on patients. So Jeff's gone deeply into the cell biology of protein folding and proteostasis, made big contributions to protein biophysics and peptide biophysics. So I'll give it to Jeff from there, and look forward to your seminar, Jeff.
So it's a distinct pleasure to be here. I think it's been 10 years since I've been to MIT Broad Whitehead, and I want to thank Matt and Amit for making this tremendous day possible. What I'd like to do today is to talk a little bit about the tafamidis story first, and then talk about some work that's emerging and hopefully will ultimately be translatable. We'll see. So, I think it's safe to say that when my lab started, the notion was that cross-beta sheet amyloid fibrils in the extracellular space drove the degeneration of tissues that didn't easily regenerate. And when we are talking about cross-beta sheet structures, it's just simply one protomer sort of per level, and then these extend to thousands of angstroms in length. So you can think about a globular protein as sort of being the size of Cambridge, and an amyloid fibril to scale would span the east coast to the west coast. So these are huge structures that disrupt certainly cellular architecture, but it was unclear whether that was really responsible for dementia and the compromise of organs, peripheral nervous system, and the autonomic nervous system. Certainly when my lab started working on transthyretin amyloidosis, I remember some of the early conversations, even at study section, And people were like, this disease affects like 20 people, so why are you working on this? Well, it turns out that now probably 5% of men over the age of 75 have wild-type transthyretin amyloidosis, and that's why this drug has become really useful. And it's arguably the third most prominent amyloid disease behind Alzheimer's and Parkinson's. The protein itself is a tetramer. The individual beta sheet-rich subunits are colored differently here. And it's the ligand-less form of transthyretin that is the form that aggregates in humans. The protein also binds thyroid hormone and binds haloretinol binding protein. And again, it's the ligand-less form that aggregates. So this is a cell non-autonomous amyloid disease. And it's one of the reasons we decided to work on it, because the tissues that are affected by the aggregation of this protein don't actually make the protein. And that gives you a lot of advantages in terms of sorting out the mechanism. So for the periphery, the liver secretes the tetramer, and when it's in the extracellular space, the tetramer dissociates, and it affords a wide variety of non-native structures, including amyloid fibrils. And it's this process of aggregation, presumably, that drives the destruction of tissues that don't make the protein. So we felt that there was enough information suggesting that aggregation was important to really spend a lot of time to figure out the mechanism of action. And given that we had a slow start today, let me just say that there's very little cell biology here, in the sense that quality control for the vast majority of the transthyretin mutants isn't a factor in the liver. So the mutants are secreted at wild-type-like levels. And of course, the patients are generally heterozygotes, so that means their tetramers are comprised of wild-type and mutant subunits. It turns out that the rate-limiting step for transthyretin amyloidosis is dissociation of the tetramer. Everything after that is relatively fast, and there's no nucleation barrier like there is in A-beta or polyglutamine or alpha-synuclein aggregation. So once you form the misfolded monomer, it's thermodynamically favored and kinetically permissible to aggregate quite fast. And of course, I mentioned the mutations. There's more mutations than there are amino acid residues that lead to the familial disease. And these mutations more or less do the same thing. They hasten tetramer dissociation, and they build up the concentration of the misfolded monomer that drives subsequent aggregation and therefore puts the patient at risk. So when Ted Foss was a graduate student in the lab, of all the pathways by which transthyretin could dissociate, he discovered that the bottom pathway is operational. So the tetramer comes apart about this dimer-dimer interface, and the dimers quickly dissociate to monomers that subsequently misfold. Now, it turns out that this is one thyroid hormone binding site, and this is a second. And these are 99% unutilized in all of us. So the idea was obvious, right? To take advantage of these unoccupied small molecule binding sites and to fashion ligands that would stabilize this dimer-dimer interface. So the idea was to stop the entire process of protein aggregation from newly synthesized transthyretin. This was a little bit of a risky strategy, because if the amyloid that was deposited was the main driver of these diseases, in principle, blocking newly synthesized transthyretin might not have a significant role in slowing disease progression. But if it was the process of aggregation from newly synthesized protein, this strategy should be useful in that we don't have to guess which one of these non-native structures is the main driver of pathology. So while we were busy at work making small molecules that stabilize the protein, a very important genetic observation was made by this woman, Teresa Callejo, in Portugal. So she was studying individuals who carried the VEL30 MET mutation. This mutation leads to polyneuropathy around age 30. And she realized that some of the big families that she was taking care of, half the kids developed the pathology, the other half didn't, even though they all carried the VEL30 MET mutation. She was smart enough to sequence them, and in fact, the kids that were protected had the 3119 MET mutation on the other allele. So they had a disease-associated mutation on one allele and an apparently protective mutation on the other allele. And of course, we reasonably guessed, since the protein was a tetramer, that the 3119 MET subunit simply thermodynamically stabilizes the tetramer. But we were wrong. When Per Hammerstrom was a postdoc in the lab, he showed quite beautifully that this was a kinetic effect, that incorporation of these interallelic trans-suppressor subunits actually increased the dissociative transition state proportional to the number of 3119 MET subunits in the tetramer. And that, of course, slows down tetramer dissociation and amyloid fibroformation, since dissociation is rate limiting for aggregation. Now, of course, as chemists, we have very little in the way of tools to engineer transition states, but we're pretty good at engineering activation barriers by fashioning small molecules that bind to the native state over the dissociative transition state, right? So the stronger the small molecule binds, the more the activation barrier is increased. So we needed a way to demonstrate that this was actually occurring in patients. And so we developed this so-called subunit exchange assay, whereby the tetramer in black is the tetramer that exists in the patient. Could be all wild-type subunits. It could be a mixture of mutant and wild-type. And then to that, we add this tag tetramer that has just a little acidic tag at the end, the flag tag, actually. And this causes tetramers incorporating the tag subunits to migrate at longer retention times on an ion exchange column. And so basically, if you have enough kinetic stabilizer around, you shouldn't see subunit exchange, and therefore the middle peaks shouldn't grow in. But the problem, of course, is that how are you going to detect transthyretin? There are 4,000 plasma proteins. So when Sunwook Choi was a postdoc in the lab, he came up with this small molecule stilbene that was not fluorescent until it reacted with the PK perturbed lysine residue, forming a covalent conjugate. That conformationally restricted the stilbene, which turns the conjugate fluorescent. So you can easily detect transthyretin in the sea of the other plasma proteome. So this was actually one of the early titrations we did in humans. We wanted to understand how much tafamidis was required to completely stabilize the roughly 5 micromolar tetramer concentration. This was actually a Nobel laureate who lived very long, in part because of tafamidis, and we sort of figured out with him that the 60 milligram dose was optimal for completely stabilizing the tetramer. So as you can see, chemically, tafamidis is pretty simple, but it fits beautifully into transthyretin and has a dissociation constant on the order of less than 10 nanomolar. And actually, this half-life is not quite right. It's well over 30 hours, which is great because if a patient misses a dose, it's not a big deal. Okay, so this is the clinical trial. This was a risky trial for sure because the neurologic impairment score has been used for all diabetic neuropathy trials, which have all failed. But the thinking was that this disease progressed a little bit faster, and indeed, you can see that the placebo group progressed along, but the tafamidis-treated group progressed more slowly on average. And this was sufficient to get approval in virtually every country except the US. And the idea was that the FDA said 3 years before the trial started, it's a brilliant idea to use these VL30 MET patients because we understand the disease. And when we brought the clinical trial to them, they said, wait a minute, this is only on one mutation, and you did the trial in Portugal, so do it again. Well, it's still not approved for polyneuropathy, which is kind of crazy, but we can talk about that. Now, the important take-home from the trial is that the tafamidis group and the placebo group are as equivalent demographically as you can get in a clinical trial. So look what happens in terms of the fraction of responders if you just delay treatment by 18 months. So you go from 70% of the patients responding to less than 50% by a slight delay. And this turns out to be true in all the trials that have been done on transthyretin amyloidosis. So treating early in these diseases is really critical. And Alnylam here in town and Ionis in San Diego have both come up with strategies to degrade the transthyretin messenger RNA. Ionis using the RAINS RNase mechanism, and of course Alnylam using RNAi. And those oligonucleotides have both been approved by a number of regulatory agencies. And again, about a third of the patients don't respond. So this suggests that in those patients, even though they're fairly early in disease progression, the proteinopathy, which probably triggers some other comorbidity, the comorbidity is now driving the pathology, such that if you fix the proteinopathy in that subset of patients, it's not sufficient. And I think this is going to be a common theme in treating amyloid diseases. And so detecting them early and getting drug on board early is really important. Now, before tafamidis came on the scene, liver transplant was used as a way to kind of replace the mutant mixed heterotetramers in plasma with a wild-type homotetramer. This procedure surgically is quite complicated because they have brittle vessels, and unfortunately 10% of young people die from the procedure. And of course, in the normal liver transplant population, that problem is way, way less than 10%. But of course, the notion that you can stabilize the protein in the periphery by doing a liver transplant actually both prolonged lifespan, as you can see from this Kaplan-Meier curve, and improved health span relative to an untreated group that's historic in this case. And then tafamidis, of course, does a very good job at extending lifespan and health span. But the reason I brought this up is that these patients, about 12 years after treatment, start developing dementia. It's pretty severe, and that's because transthyretin is also made in the brain, but it's secreted at an order of magnitude lower concentration. And remember, aggregation is concentration dependent, so it's not surprising that the CNS phenotype would be slower to develop. It's actually synthesized by the choroid plexus in the brain. And by PET imaging, after 10 years, you see very little in the way of amyloid in the brain, but after a dozen years or so, you see a lot more PET image. These patients not only develop dementia, but they also develop focal neurologic episodes, which is that you lose consciousness anywhere from a few seconds to longer, and that's obviously a huge problem in terms of being independent. Now, fortunately, although we certainly didn't design this or think about it at the time, the tafamidis brain exposure, even at the 20 milligram dose, is pretty significant. So tafamidis is shown in the gray bars, and the tetramer concentration in the brain revealed by the fluorogenic ligand that I showed you is shown in the brownish bars. And so what's kind of interesting is we've been following about 500 patients now for 14 years in Portugal, and those individuals still haven't started developing dementia. And I think this is the way to go after Parkinson's disease too, is to aggressively treat the peripheral symptoms with a blood-brain barrier permeable compound to basically prevent the CNS pathology. We'll see how long this lasts, and Of course, most patients are now not on 20 milligrams, but are on 60 milligrams for the reason that I said. Now, it wasn't, I think we were smart, and the FDA made another brilliant move. They thought that tafamidis would turn the transthyretin amyloidoses into a lysosomal storage disease, and they forced us, instead of using the 60 milligram dose, to use 20 in the first trial. That actually is completely unfounded, but that's the reason that we initially used 20. So this is a big problem in the elderly. Wild-type transthyretin aggregation, it really compromises the heart and probably the autonomic nervous system and causes cerebral amyloid angiopathy as well, like a good fraction of Alzheimer's patients have, which the vascular in the brain gets very fragile and the patients have a lot of bleeds. So Matt Maurer is a brilliant cardiologist from Columbia University. He developed the trial, and we decided to compare 20 versus 80 milligrams, which is bioequivalent to 60 milligrams of the free acid form versus placebo. The trial was pretty complicated. It was a combination of all-cause mortality and frequency of cardiovascular-related hospitalizations. This was a year worth of arguments, but fortunately Matt won, and it was a brilliant choice, actually, as you can see from the p-value in the clinical trial. So the data from the trial didn't look so great at 30 months. It was statistically highly significant, but fortunately, these groups have really started to spread out. And I'm told by Matt and others now, since you're treating these people much earlier, that this curve is really flattened out now, which is what you hope sometimes when you go from a clinical trial to the real-world population. It doesn't always work that way, but sometimes it does. And just like the data I showed you earlier, if you're in early stage of disease, the reduction in the risk of death is pretty high. If you progress to class 2 New York heart failure, then you go down to roughly 40%. Now, the last year has been an amazing year for developing pharmaceuticals for neurodegenerative diseases. Despite all this nonsense in the literature about protein aggregation being not relevant because of the early failures in Alzheimer's disease, I think the data with lecanemab is quite striking. So this came out of Lars Landfeld's lab in Sweden. He's the guy who discovered the Arctic mutation, and he developed this antibody against A-beta oligomers. Turns out in the clinic, it clears fibrils incredibly well. So at the end, we don't know, which non-native structure is the main driver, but this monoclonal antibody slows progression by 26%. Tofersen also works quite nicely. It's a RNase-mediated SOD1 degrader, and in the extension arm of the trial, it clearly slows the progression of a small subset of patients whose ALS is caused by SOD misfolding. And I've told you quite a bit about the various drugs for transthyretin amyloidosis and light chain amyloidosis. Also, the drugs prevent aggregation. So all of these compounds support the notion from clinical trials that protein aggregation drives these diseases. So what's the mechanism? Well, we don't know exactly, but you could imagine if you have a substantial amount of protein that has myriad of abnormal conformations, you'd have a lot of abnormal functions. And over decades, this changes the programming of cells and other tissues and leads to pathology. That's the current thinking, and there's a fair amount of evidence for that. But frankly, people don't like that mechanism, but I think we're going to have to live with that. We'll see. Okay, so an embarrassingly long time ago, we came up with the notion that another way to go after these diseases would be to pharmacologically manipulate the protein homeostasis network. So the notion is that as a polypeptide chain is on the ribosome, it starts to interact with chaperones and co-chaperones, and then there's a kinetic competition between folding, misfolding, and concentration-dependent aggregation. And this kinetic competition can be managed by the roughly 1,200 proteins that comprise the protein homeostasis network. So we thought that the best way to test this hypothesis was to first see whether we couldn't enhance protein degradation, biological degradation, because this would get rid of not only aggregated proteins, but a lot of these patients have aggregated RNA along with their proteins. They have lipid disequilibrium. And the lysosome, which comprises about 65 hydrolase enzymes, is really good at getting rid of all of those things, including dysfunctional organelles and so on. And so this is a process mediated both by proteins and by lipid synthesis and reorganization, where we're in a double membrane structure through receptors ultimately encases substrates to be degraded that then fuse with the lysosome to degrade those structures. And the reason that we were excited about this pathway is that the human genetics are absolutely compelling. So there are mutations in every one of the steps of autophagy that slightly compromise the efficiency of autophagy. And in humans, upon aging, these mutations lead to a wide variety of degenerative diseases, suggesting that if you could activate autophagy in the elderly, then maybe this would be quite a general strategy to intervene in neurodegenerative diseases. And this is really important because there are a lot of neurodegenerative diseases that are too small for pharmaceutical companies to get very interested in. So it'd be nice, unlike tafamidis, to have a more general strategy to treat these disorders. So ironically, nobody's done a high-throughput screen in autophagy, and we decided the best way to do that would be to feed cells oleic acid, which causes them to build up lipid droplets that can in turn be imaged by Bodipi. And then the idea is that if you have autophagy activation, you get rid of lipid droplets. If you have an inhibitor, they build up even more. And this screen actually worked really well. So we were able to prosecute over 1 million small molecules. So discovering the compounds was the easy part, as you all know. The hard part is discovering the mechanism of action. So we set the bar pretty high. We wanted to reduce lipid droplet area by 50%. We didn't want cytotoxic compounds, and the screening concentration was 5 micromolar. So there are a lot of nice assays that are a lot harder to perform in the laboratory than the papers that are published indicate. And this is one of them. So this is a ratiometric assay where the process of autophagy generates ATG4, a protease that cleaves at this interface. That affords the effector LC3, which is involved in bridging the phagophore with receptors, except that this LC3 is, has GFP associated with it, and so it can function, but of course, when GFP gets in the lysosome, it gets degraded. Unlike the right side of the molecule, where you have a mutation that prevents lipidation of LC3. And so that piece stays in the cytosol, and you can get a nice ratiometric analysis. And so those red streaks are compounds that over a 5-point dose response curve enhance autophagy over the course of 72 hours. So we ended up discovering about 30 small molecules that activate autophagy, importantly through an mTOR-independent mechanism. Now, mTOR, of course, when you inhibit it, also suppresses the immune system. That's not to say that mTOR inhibitors, which activate autophagy, aren't going to become useful drugs. I think they will. But clearly, in the elderly who are already at risk for infectious diseases, if you have an alternative, it's probably better. But Steve Haggarty's group has done spectacular work on mTOR inhibitors and shown their utility in tauopathy models and others. So there are good downstream reporters of the mTOR kinase, and so it's relatively easy to figure out whether the compound works through an mTOR inhibition mechanism. So this is rapamycin, the prototypical mTOR inhibitor. This is a compound that we're pretty excited about, A5. It does a lot of good things, as I'll show you, and it certainly doesn't work through mTOR. And mTOR has several downstream targets, and you want to look at all of them, and we have. Just don't want to bore you with that today. So Steve Finkbeiner made an amazing assay where he fused Dendra-2, which can be photoactivated to a 4-fluor, to the autophagy effector LC3. And so you can look at the rate at which this is cleaved in a pulse chase type experiment, and you can see a nice dose-dependent response for A5. This is a molecule that's different by a methyl group, and it's inactive. So we're pretty confident that you are achieving dose-dependent autophagy activation with this compound A5 that works in part through a TFEB mechanism. So somehow it activates transcriptional reprogramming that controls autophagy, but despite a tremendous amount of effort, we still aren't confident what the target is, but we're working on that. So as many of you know, axonal inclusions, as initially reported, I think, by Larry Goldstein, are prominent features of pathological specimens. And the notion is that the aggregates accumulate in the axon, they block trafficking, and of course that leads to the demise of the neurons. And so we've been collaborating with Sandra Encalada's group at Scripps, and she's interested in familial prion disease and other neurodegenerative disorders. And you can see how slow trafficking is in individuals with these octopeptide repeats that confer aggregation relative to wild-type signaling. Okay? So they published a beautiful paper in Science Advances on the mechanism by which these aggregates form. They're within multivesicular bodies in the lysosome. And again, what's really amazing about A5 is that in neurons, it's much more potent than in cancer cells. So now instead of a dose-response curve starting at 1 micromolar, we're talking about 50 nanomolar, which is, activates clearance of these endogrosomes, as they're so called, quite nicely in a dose-dependent fashion up to about 500 nanomolar. And this is another cell-based neurodegenerative disease, namely a tauopathy model that the Haggarty Lab has pioneered. And treatment with A5 nicely protects these cells against subsequent oxidative stress, excitotoxicity, and proteotoxicity. So we're really excited about this molecule. This molecule is also amazing at clearing tau. I'm just not going to show you that data today because the Haggarty Lab has really demonstrated that beautifully with rapamycin in their publication. It's effectively the same. So this is a project that probably a third of my lab is working on now because the genetics are so compelling. And I think so many people in biotech and pharma have given up because the assays are so challenging and the targets are so hard to find. But that's what we should be doing in academia, because if we can find only one of these compounds and validate it, make it blood-brain barrier permeable, we can do a clinical trial and really test this hypothesis. And if anybody's interested in helping us, we need it. Okay, so the last thing I want to talk about is the notion that the proteostasis network can fix loss-of-function diseases. And loss-of-function diseases usually occur when a mutated protein that's coming off the ribosome gets degraded by the proteasome or by the lysosome instead of folding and performing its function. So I would say the challenge with adapting the protein homeostasis network, because there's more than 1,000 components, is a priori you have no idea whether you're going to achieve enhanced folding or enhanced degradation. It depends on the substrates. So you have to empirically test this. Now, the best way to upregulate proteostasis network capacity is through upregulation of stress-responsive signaling pathways. So there's a stress-responsive signaling pathway for each proteostasis network in each subcellular compartment. Jonathan has published beautiful papers on this idea. So the unfolded protein response controls the proteostasis network capacity in the mammalian secretory pathway. So as a consequence, if we want to enhance folding and diminish degradation, then maybe what we can do is to reprogram the proteostasis network. And the idea is that since these chaperones and co-chaperones bind to things that are partially folded, you can resculpt the folding free energy diagram and drive more mutant protein to the folded state. And if we had a pharmacologic chaperone for that protein, we could, in addition, pull more protein to the folded state through native state binding, which, in fact, tafamidis is an incredibly good pharmacologic chaperone. But that probably doesn't have very much to do with the mechanism, which is why I didn't talk about it today. So the unfolded protein response has been well worked out, by Peter Walter, Cosmori, and David Rahn. And today I'm going to focus specifically on the ATF6 arm. So the notion is that when the proteome starts to misfold, BIP goes, chaperones that are bound to ATF6, like the HSP70 BIP, go to the rescue. That allows ATF6 trafficking to the Golgi, where site 1 and site 2 protease cut it, liberating the transcription factor that goes into the nucleus and upregulates somewhere between about 50 to 75 genes. Those are almost exclusively chaperones, co-chaperones, and the like. So we decided to do a pretty well-proven screening strategy where we hooked the reporter for the ATF transcription factor, so-called ERSE, to a luciferase reporter. So this is actually the regulatory sequence for the HSP70 BIP that's a prominent player in the endoplasmic reticulum for folding proteins. So we screened nearly 1 million small molecules with a cell-based reporter. This turns out to be pretty hard because at the end of the day, you have to do RNA-seq to drive the SAR. And we figured out how to do this. It's pretty crazy, but we've been doing it now for 10 years, and we have really good compounds. So compounds that work at 50 nanomolar in a wide variety of cells. You can see that some of these compounds, like, for example, well, this is actually genetic overexpression of ATF6, and Matt did this when he was a postdoc showing the specificity of this response. Pharmacologically, we never get that much enhancement. But I think that turns out to be a blessing in disguise, because a little bit goes a long way when you activate these pathways. Now, 132 is a compound that I'll show you a little bit today. It's much less selective. Matt, can I get a time check, please? What time is it?
It's 5 o'clock.
Okay, I will be done in 5 minutes. Is that okay? So
25 minutes late.
it's okay. I think I can get people on their way on time. So 147 is quite a selective ATF6 activator, and 263 is less selective. Chemically, these molecules are almost identical, and the activity of all these molecules require trafficking to the Golgi. If you inhibit site 1 or site 2 proteases, in this case by a very potent Pfizer compound, you see that these molecules are no longer useful. So it really requires ATF signaling. And these molecules are prodrugs, as it turns out. So like acetaminophen, this molecule has an amide and a phenol. And those kind of structures, when they're ortho or para, tend to be quite easily oxidized. In this case, first it forms the quinonamine, which tautomerizes to the quinone methide. And nobody in their right mind would make a quinone methide to do anything in humans because these are incredibly reactive electrophiles. But this is what sometimes comes out of a high-throughput screen, right? Turns out that this molecule integrates pretty selectively into the ER membrane. And there's a P450 in that membrane that most likely oxidizes this small molecule. And the proteins that it reacts with are almost exclusively protein disulfide isomerases. So if you compare the proteomics to all the electrophilic probes Ben has made, Ben Cravatt, you know, his hit hundreds to thousands of proteins. This guy hits a half a dozen. Now, this still scares the bejesus out of most pharmaceutical companies, right? They don't want anything to do with covalent inhibitors, although I think that's slowly changing. So we had the good fortune that we could modify one of these with an alkyne. It's still quite active in the luciferase reporter assay, and that allows you to go fishing by affinity chromatography, mass spectrometry. And at the top of the list are the protein disulfide isomerases. That made us very happy because Kosmori had implicated PDIs in the oligomerization of ATF6. And even on a fluorescently visualized gel, you can see how selective this small molecule is in terms of modifying those disulfide isomerases. So the model is fairly straightforward. The notion is that the inactive form that can't be trafficked to the Golgi are heavily disulfide linked, moderated by PDI action, and that if you inhibit the PDIs, you can get net reduction and trafficking and activation of the ATF6 arm. Despite a tremendous amount of effort, we still don't have non-covalent inhibitors for this pathway. That's not the case for IRE1 XBP splice. We have very potent non-covalent inhibitors, and I think that's going to turn out to be an incredible pathway. So Peter Walter's group made a nice inhibitor for ATF6 that stabilizes the non-trafficable quaternary structure. And in a dose-dependent fashion, this molecule sepin inactivates the activation afforded by 147. So it's exactly what you'd expect if this mechanism was operational. So we've been applying this to alpha-1 antitrypsin disease. So this is a huge misfolding disorder. The way that it's treated today is that alpha-1 antitrypsin is replaced in these patients and to make up for the deficiency. So the way that this molecule works, basically, this is alpha-1 antitrypsin. It has this so-called reactive center loop. It reacts with neutrophil elastase that, unregulated, chews up your lung and leads to a variety of disorders. And the idea is that this inhibition works by covalently cutting this activated loop. Oops, sorry. So you get quite a significant rearrangement, insertion of the reactive center loop into the beta sheet. And when you have this, everything's good. This is the way you regulate having too much elastase function and hence too much inflammation in the lung. The problem is that these mutated proteins not only are inactive, but they also polymerize by this so-called strand inserting into the neighbor's sheet. And so you end up with a domain-swapped version of the alpha-1 antitrypsin protein. And so what I want to conclude is that these are called antitrypsin deficiency diseases because there's not enough alpha-1 antitrypsin due to misfolding and aggregation. This also causes liver cancer by virtue of forming these polymers in the liver, although that's a really hard disease, I think, to do a trial on. So I'm going to focus on the lung disease today. So we just published a paper showing that ATF6 activation is really quite effective in secreting mutant forms of alpha-1 antitrypsin. So this isn't the end, but it's the beginning, right? I think it's the first conclusive demonstration that you can change the balance between mutant folding and degradation by reprogramming the protein homeostasis network. And since we started really late today, maybe I'd just show you a little bit of data and jump to the conclusion, okay? So so basically, this is a so-called trafficking index based on N-glycosylation. So N-glycans get elaborated in the Golgi, as Laura and others have beautifully shown. And you can see that treatment with the ATF6 reprogrammers enhance the amount of protein that folds and gets to the extracellular space. So I'm going to pass on this. So, and unfortunately, the polymer levels are reduced quite substantially. That's also what you'd expect if you had reprogramming of the folding versus degradation pathway. And what's really cool is that the alpha-1 antitrypsin that is secreted is monomeric, and you get a substantial increase in the monomeric concentration, and critically, it's active. So it reacts with alpha-1 antitrypsin, and remember the inhibition is conjugate formation, so it's really trivial to quantify this either by ELISA or by just standard gels, and basically you double the amount of activity in these patients. In these liver cells that are transfected with these mutants. I want to go forward now and just show you kind of the conclusion. So this paper's out, so you can read it. What Bill Balch did is basically show that this strategy works for most of the mutations that cause antitrypsin disease. And here we're looking at the amount of conjugate formation as a function of the various mutations. So, you know, I'm not absolutely sure that ATF6 is the best pathway, but it demonstrates proof of principle, and we're working hard on this. And we've recently also shown that this approach works really well for folding mutant GABA receptors. And I think this is probably the first trial we'll do because this causes seizures in kids. The trial is really straightforward, and we already have blood-brain permeable compounds that are turn out to be IRE-XBP splice activators that I haven't told you about, although they ATF6 activators work very well in addition. So not surprising, we did the IRE-XBP splice screen. Similarly, we screened for small molecules that that overexpress a given gene controlled by the IRE-XBP splice pathway. So I think I'm going to stop there and just say that, you know, it's taken us 13 years to kind of take advantage of the proteostasis network, but I think we have enough tools now. And let me say that quite surprisingly, the other thing that we found is that inhibitors of these stress-responsive signaling pathways can be equally effective in the right context. So if you change the stoichiometry of the components that make this folding versus degradation decision, that can also be a very powerful approach for the right protein. So maybe I'll stop there and we can have a discussion. Thank you.
Thanks, Jeff. That was great. And I'm sorry about the technical troubles that had us running late, but thank you for working on that with us too. Open for questions, and we'll try to run mics to people so that there's a whole bunch of people on Zoom and they'll want to hear as well. Questions? Be brave. Okay, here you go.
Hi, John.
Hi. In your first talk on the protein that you made the drug against, does inhibiting that aggregation have any effect on amyloid aggregation in the brain for patients who have amyloid mutations?
Yeah, that's an exceptional question. The answer is that amyloid in tafamidis-treated patients does get cleared slowly, but it seems to be so slow that it can explain the kinetics of the clinical response. So in the case, at least, of the transthyretin amyloid diseases, what we see go down very fast and parallels the clinical response are all the soluble aggregates that are in the blood. Those we can monitor now, and they decrease. Now, correlation doesn't mean causation. And I think the biggest problem we have in all these diseases is that we don't know what non-native structures are the main drivers of neurodegeneration.
Do you think they would have any effect in Alzheimer's?
You might repeat the.
So
Just
sorry,
repeat the.
would what have an effect in Alzheimer's? Oh no, so I suppose the only way tafamidis could perhaps have an effect in other neurodegenerative diseases if it relieved pressure on the proteostasis network, right? Freed up the lysosome to be a better degrader of, say, A beta or tau. My sense is that probably is unlikely. I mean, of course, the lecanemab data look very good to me as a starting point. You know, slowing something down by 26% is nothing to sneeze at, and we have a starting point now where we can start to add on mechanistically distinct compounds.
Yes, hi there. Really, really great talk. Thank you for sharing that with us. I got stuck very early on this V30 T119M story, very, very early, and that you mentioned. And I was just curious, kind of how many stories are you seeing like that, right? Where there's another mutation that's interacting with the mutation that you know, right, is playing a role in protein misfolding. And do you speculate that there are many other stories like that that maybe we're just not picking up because of the way that we're designing our screens for the mutations?
I mean, there are other examples of interallelic transsuppressors, but to my knowledge, not in amyloid diseases. That data was sufficient to allow the FDA to basically permit us to go straight from in vitro data humans. So they were convinced enough by that data that they didn't require an animal model, and frankly, there wasn't one. So we couldn't have done that anyway. And there still isn't one.
Okay, thank you.
Hey, Jeff, thanks so much for that. I had just a question about the last part where you were talking about, right, upregulating the network. And it just, I hope this question is on point, but you talked about your compounds hitting PDI, different PDI isoforms. And I just wondered if you looked at whether any of those are especially relevant for the results that you see, because it might be interesting if you could identify a particular PDI, because you might be able to make very specific probes for that.
That's an exceptional question, and Aranthi, our neighbor at BC, has made such compounds, and we've tested those compounds, and they certainly inhibit PDIs, as she's published, but they don't seem to activate these stress-responsive signaling pathways. So either our compound inhibits the right combination, which I think is the right answer. So we don't see proteome aggregation, which is one way to activate the UPR. So I think we've just gotten extremely lucky in getting the right distribution of PDIs to permit ATF6 trafficking.
Oh, that's cool. So the polypharmacology is actually relevant for your example.
We think so. I mean, compound 263 activates a greater number of PDIs faster and to a greater extent. And that's probably why more stress-responsive signaling pathways are activated because that induces protein aggregation in the endoplasmic reticulum.
I have a question. You touched briefly on the fact that when you activate or inhibit these stress response pathways, it's sort of just empirical whether that'll lead to more folding or more clearance of some misbehaving protein. And I'm just curious, I mean, that's very interesting and yet also frustrating. It'd be really nice if we could get to predictive principles about what's going to happen. Do you see a path for that, or you think it's going to be very hard? Or
I do see a path for that because you can use cross-linking along with pull-downs to figure out what components of the proteostasis network are particularly important for folding, say, alpha-1 antitrypsin or the GABA receptor subunit that's misfolded. So then you can discern which stress-responsive signaling pathway controls that gene and translated protein and go that route. I think that will ultimately be possible. For us, it's just faster to do it empirically because the mass spec experiments are still not fast or easy.
any other questions? David?
Jeff, do you see any differences in the virally infected cell in the protease, the pathways that might be either exploitable or differentially a problem in treating with compounds?
So I don't think we've explicitly looked at viral infections and the role of these compounds. But Judith Friedman has made some inroads towards showing that if you activate the UPR in viral infections, it's a good host mechanism to tamp down those infections. But we haven't done that work at all, so I would defer to her publications. Matt, you know that literature too. Yes, yes. Lars Plata at Vanderbilt also is published in that area.
Any other questions? All right, in the interest of time, we'll adjourn, but thanks everybody. Thanks, Jeff.